Hi, my name is Oriel Zagazeta, and my product is the effective mutations between SARS-CoV-2 variants on RNA-dependent RNA polymerase susceptibility to mutation-inducing cytosine analogs. So basically, there's these mutation-inducing cytosine analogs that are anti-COVID drugs. The specific one that I looked at in this project is Molnupiravir. And you can see from this figure here, it has the ability to bind with different nitrogenous bases, as opposed to normally A would just bind with U, G would just bind with C. Molnupiravir has the ability to create mutations in the SARS-CoV-2 genome when it tries to replicate, thus inhibiting the virus's spread within the human host. So in the emergency use authorization, when this drug was starting to be used, they found that different coronavirus strains had different susceptibility to the drug. For example, Delta was more susceptible to the drug than the Gamma variant. And the way that this drug works is it targets NSP12, shown in this picture here, within the replication transcription complex, which basically just replicates the viral genome inside the human host. So molnupiravir will pretend like it's a base, get incorporated, and then cause mutations. So the mutations between the Delta and Gamma variants can be seen on the left side of this image here with the G671S in yellow. I predicted this mutation created a difference in either folding or binding. It's also relatively close to other proteins within the replication transcription complex. But overall, this is the mutation that was observed between the two variants and what we're testing through this project. Now, these proteins aren't commercially available, and I needed to be able to make all the proteins shown earlier, NSP7, 8, 12, 13, 10, and 14. So to do this, I applicated a cloning and expression strategy using polymerase incomplete primer extension, or PIPE, as shown in this figure here. Basically doing a form of PCR that has some sticky ends on the end of the PCR products, put them together and create a recombinant plasmid that can be absorbed by E. coli for future expression, and that will have the gene of interest that we're wanting to study, along with associated tags, promoter sites, lock operons, etc. This is an example of that cloning strategy applied to NSP13 with a vector of PET101. So this process had to be replicated for all of the NSP genes that I mentioned earlier. And after that was done, it was a very lengthy process with PCR transformation, and then finally confirming all of those plasmids with sequencing. So we confirmed about 30 to 40 plasmids that were ready to be sent off for gene expression protein purification, along with different variants of NSP12 that were created to ease with folding issues that might occur when using E. coli as opposed to mammalian cells. With additionally creating the G671S mutation using an adapted site-directed mutagenesis protocol, which replicated the original primer but used original plasmid but used a mutagenic primer to create the G671S mutation. Now, before going on to large-scale purification, I tested a lot of these proteins that would ensure that the gene expression was working using small-scale experiments. As you can see here, the asterisk showed that there was a lot of expression at the target location, showing that the protein of interest was being expressed properly. At the same time, there was a little bit of bands, as you can see this figure, at a lower weight. This might show degradation of the protein of interest. It might show early termination, but overall getting those bright bands at around 110 in this case, 110 kilodaltons in this case for NSP12, showed that this was fairly optimized and we could apply this to the large scale expressions as well. So going into the large scale expression protocol, basically just amped everything up so that we could get as much protein as possible per run. And these also showed kind of similar results. We have a very strong band at 110 kilodaltons for NSP12. And then moving on to purification and isolation, I used a immobilized metal affinity chromatography column with a fast protein liquid chromatography system. This used the tags that were mentioned earlier with the vectors to isolate the protein from the other proteins that would be created by the E. coli. As you can see on the left, we got a significant elution peak for this protein, and on the right, you can see the bands are a lot more isolated as opposed to just the original gel. We have a lot more isolation and purification of this protein as opposed to previously. So this process had to be recreated for all the other proteins that need to be made. Currently, we have NSP12 all set and ready to go. We still need to do the rest of NSP7, 8, 10, 12, 10, 13, and 14. Additionally, is a development of the actual assay to measure the polymerase activity when it's exposed to molnupiravir and a control as well. And that's through a fluorometric dye assay with a quantifluor dye using an RNA hairpin substrate that will measure polymerase activity by seeing how much of the substrate is actually polymerized by this replication transcription complex. Other foundations of this project could be explored using other COVID-2 strains that have developed over the years. We could explore the Omicron strain. There's other mutations that likely have occurred within NSP12 and other associated replication transcription complexes. The replication transcription complex that could be explored and adapted the similar protocols of site-directed mutagenesis and the cloning protocols used in this experiment to explore the effects of those mutations on other mutation-inducing cytosine analogs like molnupiravir. Just like to thank members of the ESLS lab and definitely my mentor, Dr. ESLS, for helping me through this project and all the support I've received throughout the years. And these are my references. Thank you.